today we will start the chapter related to cell, and in which we are going to read about the following things, which are first the history about cell discovery and theories, then basic cell structure, then difference between prokaryotes and eukaryotes, then similarities as well between prokaryotes and eukaryotes, then properties of prokaryotes, properties of eukaryotes, then cell membrane, which are common in both of them. Then cell wall, which are different, endomembrane system, mitochrome, plastid, ribosome, cytoskeleton system, cilia and flagella, centrosome and centrioles, nucleus and mitochondria. So there are few things which I want to tell you that in Imat, like I said earlier, that we will follow two books. In these two books, there are first is NCERT from India, NCERT Biology from India. Why we are using it? Because I used it. As simple as that. I used it. It is a very good book. There are only enough details provided, which are in a story, a complex story. It is like a puzzle. And after that, A level biology by Cambridge. So here in them, which are we going to prefer in which cases? So in case of NCERT, it will give you information, and in case of A level, which also gives information, and in this case the information is like it is very well, how do I say that word, it is divided in systematic way. But in NCRT there are paragraphs and you have to read them. Okay, but I will teach you from NCRT to, because I know it. Okay? So there should be no issues. But in A level, I think because it is divided in a systematic way, so then you can learn about how things are going to work, how there are specific things in a specific area, in a specific organ. You can know better from it, because if you read the same thing from NCRT, there is one point about a particular topic in a paragraph A, and one topic you will find, one point about the topic you will find it in last of the chapter. That's why A level is also preferred. So I think we should start now, and if you have any query you can just ask me. I will drop my contact. Probably I will not drop the number, but I will drop my contact like Telegram ID, and maybe my Discord, and my email as well. So you can contact me anytime you want. Okay. So I think we should start. So if you talk about cell, what is cell? It is the first thing which should come in our mind. So if you talk about what is a cell, then cell is a unicellular organism which is capable of, first thing, it can live independently. Independently living means it do not need anyone else helps. Like if you talk about in a cell, what are the things in a cell? Like there's a nucleus. It requires ATP production. It requires ATP, GTP, and stuff, which are not formed in the nucleus. They come from outside, from like ATP. ATP from where does the ATP come? ATP comes from mitochondria. So nucleus is not an independent living cell. If you talk about ribosome, it is not an independently living thing. Okay. So what do you need in a cell? It should have it must have independent existence. Okay. It can interact with its environment and it can complete its life processes. After that, there comes the performing the essential functions of life. Especially, I mean, no, of course, if you want to live independently, then you should do some tasks which are essential for your life. Like if you are a student and you want to live independently, then what should you do? You should do part-time job. Why? Because you want to have money. If you can have money, you can pay for your rents, you can buy food sometime. If you are living independently, you should know how to prepare your own food. Okay, you understand what I'm saying. Now in NCRT we will not cover, of course not, we are not going to cover every single line, because I will tell you some important points. After that you should go uh, Maybe you can make notes while watching the lecture, or you can go and do some questions from website like Locomotive and stuff, which I told you before in the earlier video, and you can attempt the previous year test. But I will not. I will try to make the lecture shorter, maybe like an half an hour. Maybe. So if it okay, anything less than a complete cell cannot have a independent living, which is right, because if you can't perform the essential function, you can't have an independent life. If you can't earn by yourself, you can't be independent. If you can't make your own food, you can't be independent. You have to be dependent on other person. So hence, cell is the fundamental structure and functional unit of life. It is a fundamental structural and functional unit of life. It is the fundamental. Fundamental is basic, structural and functional unit of life. Okay. So here are three scientists which are we are going to know. First is Anton von Leeuwenhoek, who was the first person to define a living cell. He was the first guy to observe a living cell. Then Robert Brown, he was the first guy to discover the nucleus, and one more guy who is Robert Hooke, who first time saw a cell, which was not a living cell, it was a dead cell. Okay, and after Microsoft invention, things got a lot advanced. Now we will talk about cell theory. In cell theory, what we are going to remember, that there were two scientists initially who gave their ideas, which were Matthias Skaldian, who was a German botanist in 1838. He studies a large number of plant cells, and he told that different kind of cells come together make tissue of the plants. Okay, and at the same time, almost in 1839, Theodor Swan, a German zoologist, he studied different type of animal cell and reported that cell has a thin outer layer, which is today known as plasma membrane. He told us about what he told us. Swan told us about plasma membrane. Wait, let me fix it. Yeah, he told us about plasma membrane, and he also told it whose base of study on his own. Okay, he told that sorry, on basis of his study on the plant tissue, he told that the presence of cell wall is unique feature of plant cell. Cell wall are not present in animals. And Swan also proposed the hypothesis that an organism consists of cell and its products. And Scalding and Swan later, what they did, they together formulated a cell story, but they couldn't tell that how does new cell arrive. Then Rolf Virchow in 1855 he told us about unicellular, acellular, and then the final cell theory, which was all living organism consist of cell and its product, and all cell arise from pre-existing cell. So of course, there are you do not remember need to remember. I think the scientists did their name in case of Imat. Imat do not go too deep into history. So here we will talk about an overview of cell. We will talk about a cell. Okay, so I think I am going to show this on a page. Wait. So what happens in a cell? So first, let me do it. Like how we decided. History about cell discoveries. It is done. Now basic cell structures. Okay, so basic structure of cell. Now we are going to learn about it, which we will do for first. What type of cell it is? So it is a prokaryotic cell. It is a prokaryotic cell. So we will go from outside to inside. So outside there is a bacterial flagella. So in flagella it is not present in every prokaryotic cell, and if it is present, then the cell is motile. Okay, after that we go inside here. It is the what is this region is called the cell membrane. If we no, cell membrane complex, I think cell membrane complex. So in case of this, there are three layers, which are first one is from outside to inside, which is glycocalyx. So it is glycocalyx whose rigid form is known as capsule, and whose smooth or we say loose foam is known as slime layer. After glycocalyx, what comes? After glycocalyx, it comes the middle layer, and in the middle layer, here comes the cell wall, which is formed of peptidoglycans, and after that, the innermost is the plasma membrane, which is same as the eukaryote, so we will study their structure later. After that, there comes the ribosomes, which do protein synthesis. Mesosomes, in mesosome we also do the ATP synthesis. They function like mitochondria. And the DNA, so DNA, in case it is in the region called nucleoid, there is no specific nucleus. DNA is just bounded. DNA is just bounded and by the positive charge proteins, and in the region it is called nucleus. After that, we goes for the eukaryotic cell structure. In eukaryotic cell structure, which type of cell it is? It seems to be what type of cell? It is a plant cell. Plant cell, we will go from again outside to inside. This is cell wall. This is cell wall, the outer layer. This one is cell wall. After that, this inner one, what it is? It should be what? Plasma membrane. Plasma membrane. After that, here this region is the cytoplasm, and these little bodies, what are they? They are lysosomes. Lysosomes. And these, what are these? They are Golgi apparatus. This phase is transphase. This phase is cis phase. And these, what are these? These are endoplasmic reticulum. Which type of endoplasmic? Smooth endoplasmic reticulum. What are these? They are rough endoplasmic reticulum. Why? Because it has these small ribosomes. And what about this? These are mitochondria. This is the vacuole. This is a vacuole. And here the middle lamella. Middle lamella is a layer. So I messed up a little bit. I messed up. So here, the plasma membrane. What is plasma membrane? This inner layer. And this is the cell wall. This is the cell wall. This one is also the cell wall, but of another cell. And here this line, this is the middle lamella, the layer between two cell walls. Okay, and this is the plasmadesmata. What does plasma do? Connection between two plant cells. Okay, sorry for bad handwriting. Okay, so after this, what are we going to do? It was the basic cell structure. In case of animals, okay, I should teach it. Okay. In case of animal cell, what is this? This is the what is this? This is the plasma membrane. Okay, the plasma membrane. Okay, after that we go inside. This is cytoplasm. After that, this is mitochondria. What is this? These are lysosomes. Oh, sorry, these are not lysosomes. These are ribosomes here. I'm sorry. The bigger ones. These are lysosomes. Here, these are mitochondria. What is this? This is Golgi apparatus. What is this? It is RER. And what is this? It is SER, smooth endoplasmic reticulum, because there are no ribosomes. Nice. And this is nucleus. These are nuclear pores. What is this? This is nucleoid. Okay. Now let's move further. Where should we start? We should start from outside to inside, outside to inside, outside to inside. So first, the cell envelope. Cell envelope. So in prokaryotes, cell envelope is consists of three layers, which are glycocalyx, cell wall, and plasma membrane. And in case of eukaryotes, it consists of, in case of plants, it is cell wall and plasma membrane. In animals, it is only plasma membrane. Okay, the cell wall, it's different. Okay, the cell wall in both of them, they are different. Glycocalyx is not present in eukaryotic. Okay, and plasma membrane, it is same on both of them, which we will learn later. After that comes the cytoplasm. Cytoplasm, which is same. Cytoplasm, same, present in both of them. Sorry. Cytoplasm, it is present in both of them. Now here comes ribosome. Ribosome in prokaryotes, there are only 70 S ribosomes. In case of eukaryotic cell, ribosomes, they are of both types. Both means 70S as well as 80S. Okay. Now here comes mitochondria. It is absent. In eukaryotes, mitochondria, it is present. It do the ATP synthesis. Okay. Here there are mesosomes. What does mesosomes do? Mesosomes do the ATP synthesis. Other two as well. Okay. And here there are no mesosomes. Mesosomes are absent. Now endomembranous system, which is absent here. It is endomembranous system. It is present. What does it do? It do function of the cell in a very coordinated way, better than prokaryotic cell. Prokaryotic cell, they are smaller in size. They are larger in size. Okay. In case of prokaryotes, they divide faster because they are smaller. Okay. They divide faster. They divide slow as compared to them, obviously. Okay. Now what else we should do? What else we should do? Their DNA. In case of them, the DNA, it is less founded as compared to the eukaryotic, as well as it is stored in a region called nucleate. Nucleus is absent. So now if you are going to say that, hey brother, the nucleate is present in the prokaryotic cell, I mean it replaces the nucleus. So no. What is nucleus? Nucleus is a structure. Nucleate is a region. Okay. Nucleate is a structure. It has its own dimension, its own boundaries. But nucleate is just a region. Okay. So in case of eukaryotes, there is a nucleus present in which DNA is present. Okay. Now there are, for movement, it has flagella, which is different from the one in eukaryotes. Okay. And here in movement, it has flagella, which are obviously different. Also cilia, which are not present in the prokaryotes. Also it has other structures like pilia, which are made of pillin protein, and it do not have that. And it also have fimbriae, which are not present in eukaryotes, obviously. Also there are other things about which we will discuss later, okay, when we will study them in details. And there are no membrane bound organelles. It is a very important one. I should not have forgotten that in prokaryotes don't have membrane bound organelles, and it have them. Okay, what membrane bound organelles? Membrane bound organelles are like the organelles which have membrane around them, which are whose content is fixed in a specific area, in a specific organ, but prokaryotic cells don't have those. About others we will discuss later in the video, because I want to complete it as well, and it will be like maybe an hour, I think. It is done. I think it is done as well. If not, we'll discuss it later. Don't get bothered with it now. Let's start prokaryotic cells. So let's start the prokaryotic cell. So here prokaryotic cell have different types of cell, which are like bacteria, blue-green algae, and PPLO, which is pleuropneumonia-like organism. Pleuropneumonia-like organisms. Okay, and like mycoplasma. What is mycoplasma? It is a type of organism which do not have cell wall, and they are the smallest. Okay, and the shapes in them are like bacillus, which is rod like, coccus, which is spherical, vibrio, which is coma, and spiral, which is spiral type shapes. Okay, and in case of the bacillus is like example as E. coli, coccus, sorry. In coccus it has staphylococci. In vibrio we have vibrio chlorella, which causes chlorella, and the cytoplasm which is present in them, which is a semi-fluid matrix, and they don't have well-defined nucleus, which is so its function is done by nucleate region. Okay, and in the DNA they have two type of DNA. They have two type. One is genomic, which carries the genetic information of cell, basic genetic information of cell. Okay, basic genetic. Okay, and other, they also have a phenotypic DNA, or we say a plasmic DNA, which gives certain phenotypic character to the cell. This plasmic DNA, they also helps scientists to study about bacterial transformation, which is essential in biotechnology, which we will study soon. Okay, not soon, I mean it will take time. It is maybe 28th lecture, I don't know, I mean maybe. Let's leave this topic here. And thus pollodine is called as plasmids or plasmid DNA, and it has certain phenotypic characters like resistance to antibiotics, and it is used to study about bacterial transformation. The nuclear membrane is found in, it is about eukaryotes. Okay, so prokaryotes, they do not have nuclear membrane, and prokaryotes have something special in them, which in the form of inclusions. Prokaryotes have infolding in their cell membrane, which are known as mesosomes. Okay, and in case of them, mesosome, which is formed by the extension of plasma membrane into the cell. It means that if this is the cell, then they are extension inside the cell of the plasma membrane. Okay, it is not a mitochondria, which I just drew. And they have three forms, like vesicles, like tubules, and like lamellae. Okay, they help in various processes like cell wall formation, DNA replication, distribution in the daughter cells, and they also help in respiration, secretion processes to increase the surface area of plasma membrane and the enzymatic content. So when they increase the surface area of plasma membrane, what does they also increase? They increase the rate of respiration. Okay, so this process, the surface area of plasma membrane, it directly helps the respiration. Okay, and also the enzymatic content. If the surface area increases, more enzymes are there, respiration is done in a good at a good rate, and also secretion processes occur at a very good rate, because they all of them are directly proportional to surface area of the plasma membrane. And in some prokaryotes like cyanobacteria, there are other membranous extensions in the cytoplasm which are known as chromatophore. What does they do? They have pigments just like plant cells, so they helps in carrying photosynthesis. Now in case of bacterial cell, if we talk about motility or non-motility, so if they are motile, they have flagella, and if they are non-motile, they don't have flagella. If motile, they have thin filamentous extension. What are flagella? They are filamentous extension. Okay, they have three parts: filament, hook, and basal body. So it is like a basal body. Okay, and here, sorry, it is a basal body, and sorry, it is a plasma membrane. I'm sorry, it is cell surface. Word slips. It's my first time teaching after a very long time. And here, what is this inside the cell surface? It is basal body. And here it is the hook. It is flexible and it attaches the basal body to the plasma membrane, sorry, to the flagella's filament. It is filament. What does it helps in? It helps in the locomotion. Filament is the longest portion extend from cell surface to the outside. Beside flagella, it also have pili or fimbriae. They don't have any role in motility. The fly, it is an elongated tubular structure made of special protein. What is the special protein called? It is called pilot protein. And what is its function? It helps the cell to transfer the plasmic DNA from one cell to another. And if I remember it correctly, it is the F plasmid DNA, which is in which case the transfer takes place. After that come the fimbriae. Fimbriae, they are small bristle-like structures, fibers sprouting out of the cell. In some bacteria, they are also helped in attach it to rocks in stems and as well to host tissues. Okay, now there comes the ribosomes and inclusion bodies. Here we will only study about ribosome. In this case, we are only going to study about 70S ribosome. 70S. Why 70S? Because in prokaryotes only this is present, and the inclusion bodies. So the dimension are like 15 to 15-20 nanometers. Okay, and they are made of two subunits, which are 50S and 30S. What is S here? S is sedimentation unit. It is an indirect measure of density. Okay, they are the site of protein synthesis. They are present in the ribosome and also attached to plasma membrane. And what does they do? Several ribosomes, they attach to the single mRNA, and then they form a chain known as polyribosome or polysome. And the ribosome polysome, what does they do? They translate mRNA information to proteins. Okay, and now there comes the inclusion bodies. What are these? These are the area where the reserve for material prokaryotes are stored in the form of inclusion bodies. So it is not organelle. It is when prokaryotic cells stores the reserve food material. what does they form? They form a inclusion body, so it is not a specific organ in which they take the food. They just make it like a spot where food is collected. They are not bound by any membrane system. Okay, they are not a membrane bound organelles. They live really in cytoplasm. Example like phosphate granules, cyanophysin granules, glycogen granules. And there are also gas vacuoles in case of what? Blue green algae and purple and green photo, sorry, not algae, blue green purple and green photosynthetic bacteria.